So what happens is, we have this population. If Hardy-Weinberg, if this population was in equilibrium, we would see these same percentages, 25% for each 4 of these different colored shapes, again in the next generation. But what we see is the blue triangle happens to mate more frequently and get their genetic pool out there. So we actually see that these numbers start to change. We now see as the new population has 40% of the blue triangle, which means that all of the other colors and shapes would be less. So this is what happens with non-random mating. Where do we see this in the world? So there was a immigrant descendant named Arnold in South Africa. He had 7 wives. There are still 356 living descendants of this gentleman named Arnold. And we know this because there's an increase in the frequency of a dominant dental disorder in this population of his descendants. So this was non-random mating. He was choosing women from a pool that lived very near him, 7 wives. He was very active in the breeding population. But let's go one step further. How many of you have heard of Genghis Khan? He was a Mongolian warrior. His rate of Asian women led to 16 million men living today sharing his Y chromosome. Oh, wow. Very prolific gentleman. So here you see some of the effects of non-random mating.
It's just a random question, so are there a bunch of rapists now? You know, they just like everything, they're collecting your genetics.
Well, not in terms of rape, but, okay, what about men who donate their sperm to fertility clinics? So there's the potential there of less diversity, depending on if a number of women go and choose vial number 007, as in James Bond.
The people.
Right, so it's going to lead to a lessening of the diversity. Your book goes into a lot more detail. I just gave you the bare minimum facts, so I'm going to do that. So we've seen this term before, consanguinity, and this is marriage between blood relatives. This typically, again, happens in royal families, or I've got another example here about the Amish, who also tend to marry amongst their own little population. We also have something called endogamy, which is marriage within a community. So those are just additional examples. So that's non-random mating. That is one of the 5 that you will need to tell me what they are. 4 factors that will lead to non-Hardy-Weinberg equilibrium. Factors that lead to changes in allele and genotype frequency. There's 5 of them. We just covered 1. The second is migration. How many of you jump on a plane and go somewhere? How many of you have ever lived anywhere else? Few of you. So this is essentially migration. I've lived in Gainesville, Florida. I've lived in Fort Worth, Texas. I've lived in Baltimore, Maryland. I'm now living in New Orleans, Louisiana. So a little bit of migration. So what happens during migration is you have the addition or removal of alleles altering genotypic frequency. If I picked up and moved and began to mate and live in Luanda, Angola, which is where I spent about a month of my time, say I really liked it there, I found somebody that I wanted to settle down with, I would be introducing pretty European alleles into that population. So again, they would be picking up some of my genetic diversity. So that, again, this is going to be addition or removal of alleles altering the genotypic frequency. We end up seeing the genetic effects of those migrations reflected in current populations. This is how we are able to go back and determine ancestor patterns and how our ancestors moved across the globe and eventually landed somewhere. So again, these can be mapped across either geographical or linguistic regions. So how hard is it to maybe find a future mate if they don't speak your language? Pretty difficult, right? So we can also kind of decide and see where those language barriers may be. So again, here's our original population, 25% for each of the shapes and colors. During the migration of this population, they run into a couple more yellows and reds, and they want to join in and migrate with the original population. And what happens is we end up with more diversity than we had before because we have higher numbers of red and yellow than we did in that original population. So our genotype frequencies are going to end up changing. They're not going to stay the same. If they stayed the same, it would be equilibrium. Climbs, which is one of your vocabulary words. These are going to be gradients that we are able to track in allele frequency in neighboring populations of individuals. And again, these will be used to detect patterns of migration. So your example in your textbook, they look at a particular recessive disorder called galactokinase deficiency, and this causes blindness. And what they're able to see is that it varies in prevalence. If you can see the green and kind of light green areas, they vary in their prevalence across Europe. And so they're able to see that it was most common in Bulgaria, but they know that some of these gypsies moved and donated that recessive allele into the populations of Austria, Germany, and to some extent Switzerland. So they're able to track that just looking at this one autosomal recessive disorder. And again, these climbs can be either abrupt or gradual, as I just showed you. These boundaries can be correlated to historical events. How many of you know about the famine and some of what led to immigrants pouring in the Statue of Liberty in New York City? New York City has a very huge melting pot of individuals from everywhere, and this is part of what happened was this historical event led to individuals pouring in. Cultural differences, and then we also talked about, we talked about language barriers, but there's also geographical barriers. So mountains and water in your way. Obviously, you can't walk to Africa, right? Because there's a great huge swath of ocean in your way. So water and mountains can also form geographical barriers to the genetic pool moving in language signals. And again, we can use all of this to determine patterns of migration. Okay, non-random mating, migration. Number 3 is genetic drift. Genetic drift is just what the word says. It is the change in allele frequency when a small group is going to separate from a larger whole or reproduces only amongst themselves. So the example here is Amish. They have essentially broken off from mainly the population in Pennsylvania, although they are in different places in the north. But they broke themselves off and they decided that they weren't going to mate with this larger population. They were going to keep mating within their own little barrier. So this changes allele frequency. These allele frequency changes are going to be unpredictable because they can be essentially caused by random sampling errors. And again, genetic drift is going to be more pronounced in these smaller populations of individuals. And that's why I chose the Amish for an example here, because they are an extremely small population within the larger population of the United States. So how does genetic drift work? Again, we're starting with our original 25% of each of the shapes and colors there. When we drift, we've decided that we're not all going to be mating with each other. So half of the population decides they want out. So the original population is left on their own, which leads to drastic changes in their frequencies here. You can see yellow and green. So what probably happened was a loss of what two shades or colors? What two colors went down? Red and blue. So the red and blue decided we're too good, we're going to go and populate somewhere else, and left the yellow and green behind. Maybe one or two outliers decided to stay behind, and that too progressively creates this next population. What might end up happening in the next generation? They'll eventually start to fade out, right? The more yellow and green we get, potentially. So what are some events that lead to genetic drift? Founding new populations. So again, this is essentially the Amish population effect. This is known as the founder effect. So you have a group of individuals that are going to essentially found an area where they want to populate and create a new population. We also have things occur like bottlenecks. So this will mean, and I'm going to go through all of these in a little bit more detail, natural disasters that occur, famine. What about the influence of human beings on endangered animal species? Right? We are essentially taking away potential mates for these animals and lowering the genetic diversity potential. Cultural separation. This doesn't happen as much now in some cultures as it does in others, but for the most part, you look at it in history and you see where, boy, they outcast you. They can get killed in some countries still for choosing the wrong mate. So when you think about these things, these are all contributing to genetic effect. The founder effect. This is going to occur when a small group is going to leave home and they found new settlements. So if you go back in your world history and you look at those small populations of men who said, hey, we're crossing the ocean. They were founders, right? They ended up landing in a certain spot and they created new populations and new settlements. The new colony of people are going to have different allele frequencies than the original population. So if you look at our settlers that came from England, perhaps, they don't still have the same frequencies that the English population has, because they're going to come here and they may start mating with the natives. And that creates a different frequency. If either it may by chance, if they lack some alleles or they have a high frequency of others, we're going to end up seeing that in that new population. And again, some of these are going to have disorders and traits linked to them so that we can track them over time. So your book gives this example for founder effect. So there's a very rare disorder that leads to severe migraine. And this is very common among these descendants in Norfolk Island and from the Pitcairn Islands. And what happened was there were 18 sailors. They mutinied. They decided they were going to leave the Royal Navy ship. And they mutinied to the Pitcairn Islands and also to Tahiti, which is here. And then they had children with the native women. And we know from their descendants, who are now in Norfolk Island, that they have essentially passed on this trait that leads to severe migraine. So this is the founder effect. How many of you have heard of mutiny on the Bounty? This is what it was based on, these 18 sailors that left. So here are some more examples that your book gives you of different founder populations that were started, the number of generations that they have gone through now, and their population sizes today. So you look at Japan. It was essentially founded with 1,000 people. Look at that. 120 million people. So you can see the effect of being very prolific in your reading. A couple of other examples. French Canadians of Quebec, and also, the descendants of the German immigrants in Germantown, Pennsylvania. But again, I like the other examples better, so just read in your book if you're having any difficulty understanding some of the effects of these different things. So genetic drift and non-random mating are the two that we've already talked about, right? So small population size is going to increase the probability of homozygosity. Y'all remember talking about homozygosity and what that means? Yes, no, maybe?
No.
What does homozygous mean? Same. What does heterozygous mean? Dr. Jaleesa. Homo or heterozygous? Okay, so what happens is you will end up becoming more homozygous over time and you're essentially going to eliminate anyone who is heterozygous because these alleles, one of these alleles will end up being preferentially selected. So y'all are clear on homo versus heterozygous. So what happens is we're going to increase sometimes the recessive phenotypes in the population. For example, we've already talked about the Amish population in Pennsylvania. Again, they're going to marry predominantly within their own community. They maintain their original small genetic pool, and what we see with them is an increased incidence of otherwise rare traits. Because of this occurrence, they're essentially getting rid of their heterozygous. So your book talked about this particular syndrome called Ellis-Van Creveld. This ends up with dwarfism, extra fingers, and heart disease, very common amongst the Amish. And you can actually see her little extra fingers there in the picture. Okay, so any questions now on non-random mating, migration, and genetic drift? No? Y'all get it? Okay, bottleneck. We have a large population. We essentially use a funnel. We drastically cut that population down, and it rebounds in population size occur with descendants of a limited number of survivors. Look at the Jews from Auschwitz. That we created a bottleneck, right? We eliminated almost an entire group of individuals so that only a small group remained. And what happened then was that only the alleles that those individuals had were able to be reproduced, right? So you can think of that as an example. So the new population is going to have a much more restricted gene pool than that original ancestral population. Your book uses the example, too, with the cheetah as an endangered species. So, say, for example, we see A through Z. We see 26 alleles in the cheetah family. When we start killing them off for their fur, for their meat, only a few will remain. We'll find out in a minute they're probably the strongest, right? They only have 4 alleles left in this small population of cheetahs. When this group starts populating again, humans back off and we say, we make some laws. The cheetah is about to extinct. We've got to leave them alone. We let them start growing again. We'll never get the diversity that we had from that original population. Those alleles are gone. All we'll have is diversity with these 4 alleles. Does that make sense? So again, we talked about the Jews. We talked about cheetahs. There was also an example of a bottleneck created by a natural disaster by a typhoon among the Tungalapese people of Micronesia. Your book gives you a few more. What the disorders prevalent among the Jewish population are. I'm not going to ask you to know this, but again, this is just for your own self. We are coming to the end of the semester. I don't know how many of you have noticed that, but that means that we're getting closer to when research papers are due, right? Yeah. I think I talk about this in every lecture now. So make sure that you've got your topic and make sure you're working on that paper, because that's a big 20% of your grade. You know, I want to see you guys do well. So if anybody's doing anything on any of these diseases listed here, you might find some more information. All right, mutations. We've already covered this in one of our chapters, right? So now we're saying it twice. So mutation stuff is going to be important to know for these again. So mutations are going to be a major and continual source of genetic variation in populations. It can introduce new alleles. It can convert one allele to another. Mutation has a minor impact, and most are silent unless they're coupled with another effect, such as small population size or selection. So we won't necessarily see a huge impact from mutations because, again, they're rare. Why are they rare? What was one of the other things we talked about with regards to mutations? Talked about the ways that our human body repairs DNA damage, right? So that's why mutations are rare. Y'all need to know DNA repair for the exam, part of your one chapter. So how do mutations create changes? So again, we start off with our original population with our 4. Mutations can change alleles. So say, for example, our blue triangle changes back into a yellow square, and our green diamond changes into a yellow square. Well, that essentially puts that diversity back out. So now we end up with much more yellow. Half of our population is now yellow. So that's a pretty big change and a big impact from the mutation here. But your book's using an example of something big to show you the effect it can have. But again, know that it's really a minor impact from mutation. We'll get to selection in just a moment, but selection usually eliminates deleterious or harmful alleles. And it does that from either not allowing those individuals with those harmful alleles to reproduce, or it kills those people off. However, we have seen some harmful recessive alleles being maintained in heterozygotes, and these can be reintroduced by mutation. I took that off of your vocabulary, this term here, genetic load, but it's the collection of all the recessive alleles present in a population. But I'm not going to have you know that one. I feel like you've got enough information to know. All right, let's get to natural selection. You've all heard about natural selection at this point, yes? So it's the differential survival and reproduction of individuals with a particular genotype or phenotype. They're not necessarily the fittest individuals. We can have selection for traits that would otherwise be bad. So negative selection would be banishment of a dangerous trait. Positive selection is going to be retaining an advantageous trait. But we also do see sometimes that there's selection for a particular allele, which I'll give you some examples of that in just a moment. So both positive and negative selection are going to lead to changes in allele frequency. So again, negative, we're getting rid of the bad. Positive, we're keeping something that's really good. So natural selection. Got our original population here. And let's say yellow square has some kind of mutation or disorder where they can't reproduce. Well, you already know they're not going to contribute, right? They're not going to contribute their alleles to the next. So the next population is only going to be made up of the blue, green, and red. So that's selection. Is that positive or negative? Negative? Why is it negative? It's banishing essentially people who can't reproduce. We see something called artificial selection. We see this every day with going to the grocery store. You think that it's not happening, but it is. Your tomatoes are being grown to be kiwis. Your corn's being grown to be sweeter. Honeycrisp apples. This is going to be controlled breeding with the intent of perpetuating individuals with a particular phenotype. But it also occurs in plants. We also do it with pets. Due to artificial selection and inbreeding of dogs, body size between two different dogs varies more than it does in any other terrestrial species. We have little teacup dogs. We have great big huge dogs. But they're all still part of the same species of dog. Okay? Or the same genus. So, artificial selection has had that much of an effect on a household pet. When you think about it, it's pretty crazy. So observations of the artificial selection were key to Charles Darwin's understanding of natural selection. And artificial selection among domestic animals decreases their genetic diversity. So what we're seeing is less diversity among our animals. So how many of you have had a purebred animal in your life? Anybody? What typically happens to your pets when they're purebred?
You gotta watch them like they didn't have diseases or
They get
anything
sick.
there, yeah.
They get sick. So why do y'all think cancer is prevalent in boxers? That's due to inbreeding of boxers. Certain types of the blind. Poodles usually have blindness and deafness because of inbreeding. So those are typically some recessive traits. We're losing our heterozygosity and becoming more homozygous due to all of that. Okay, so you know I don't make you know diseases, but I got one more I want you to know. What was the first one that I need you to know from a previous chapter? Sickle cell. So I'm going to have you know sickle cell. I'm also going to have you know just a little wee bit about tuberculosis. So natural selection can be seen in the appearance or return of infectious disease. So the bacterium that causes tuberculosis, which is also known as TB, this was first identified by Robert Koch in 1882. And TB infections have historically swept across populations, killing masses of people. Natural selection operating on both the bacterial and on the human population has lessened the virulence of this infection. It's no longer as deadly as it used to be, right? We all know that. How many of you in here have been tested for TB? All the time. All the time. Don't go there. We're not going to scare you. So what happened was, some people inherited resistance. Their parents lived, something funny? What's funny? You got a little grin on your face. I was wondering what's funny. Share. Some people inherited a resistance to it. So that helped to make it less virulent. We then had the effect of the most virulent bacteria. They killed people. They weren't able to spread it, which meant the most virulent bacteria started to die off because the people that they were infecting weren't spreading it to other people as much. So we ended up with the deadliest bacteria essentially being selected out of the population. That's an example of negative selection. And the people who inherited that resistance to TB were positively selected. So you had two events, positive selection and negative selection, which have essentially changed the occurrence of TB. Make sense? So what else does that mean? So it used to be a horrible systemic infection. Now it's essentially a severe chronic lung infection. It has made a recent resurgence. And the reasons are also a result of natural selection and mutation. The resurgence is going to reflect HIV positive and AIDS individuals and increasing bacterial resistance to antibiotics. Because since the 1980s, we have been using antibiotics a lot in this country, right? So what did AIDS do? It essentially provided millions of people with vulnerable lungs. Because again, remember, TB is a chronic lung infection. It's going to attack the lung. So you've now got another population full of people that TB can hit. In fact, a third of all people who are HIV positive have tuberculosis. We also see that the bacterial populations have been soaring. They've been generating mutations. Those mutations have allowed them to become resistant to antibiotics, which is a bad thing. So that's essentially what I want y'all to know about tuberculosis. We next have something called a balanced polymorphism. This is going to exist in populations when heterozygotes carrying a disease-causing allele enjoy a health advantage. You all know an example of this, but you're probably not thinking about it right now. It's also called heterozygote advantage, and they can have a reproductive advantage under certain conditions. So let's give you the example of one of these, sickle cell disease. How many of you in here knew that if you have sickle cell, you'll essentially never get malaria, right? That's an example of a balanced polymorphism. I can see a multiple choice test question, and I promise it'll only have sickle cell and then some random diseases that aren't on this list. For you to choose an example of a disease that is balanced polymorphism. The balanced polymorphism thinks sickle cell. So what happens is, you all know about sickle cell, because you guys know it's a test anyway, so it's like a perfect example to use, right? Remember that beta hemoglobin gene is what's going to produce sickle cell. The sickle cell causes the recessive sickle cell anemia trait when homozygous, and it's therefore under negative selection. So sickle cell allele is going to help protect people who are heterozygous from malaria under positive selection. So I'm not going to make you go into that much detail. I just want you to know that sickle cell is an example of balanced polymorphism because it does provide that health benefit. All right, they're on here again. Another slide with those 5 things: non-random mating, migration, genetic drift, mutation, and natural selection. Your book's giving you a table. It's giving you some examples. You don't have to know these examples, but I want you to look through these examples to make these 5 things make sense to you. So that if I ask you to give me an example, it's not you to. Okay? Just make it make sense. Whatever you need to do. If that includes coming to my office, come see me. Your book also shows it this way. So again, some examples of what's happening with genetic drift, migration, non-random mating, natural selection, and mutation. The same smaller examples we've been looking at for tonight. Last topic. There's something called eugenics. How many of you have ever heard of eugenics before? This was a word coined in the 1880s by Sir Francis Galton to mean good in birth. And on a societal level, it's essentially the control of human reproduction with the intent of changing a population's genetic structure. We've already mentioned tonight an example of this. How many of you could tell me what that example is? What was Adolf Hitler trying to do? Right. He thought they were good at birth. Eugenics applies to situations like that. You have a population who says, we're better, let's try to eliminate that population of people. So positive eugenics would promote reproduction among those considered superior, whereas negative eugenics would interfere with the reproduction of those judged to be inferior. So this is one more last vocabulary word to know is eugenics. I'm not going to make you tell me when and by whom at this point, but I might ask you what that term means. One more little thing on that. So some people consider what we do now, genetic screening, to be eugenics, because essentially we are screening for a better genotype, right? Because we want to make sure that our children don't inherit disease. But essentially what we are doing is we're aiming to prevent or alleviate human suffering for those who come after us. All right, there's your vocabulary for tonight, not too many. Pretty brief chapter on that. And